Hi, my name is Abigail Koppes and I'm a student at Rensselaer Polytechnic Institute working with Dr. Deanna Thompson in the Neural Engineering Lab. So my project today is discussing the use of the biophysical cue of electrical stimulation to help guide neurite outgrowth following a peripheral nerve injury. So during a peripheral nerve injury, you have axonal degeneration with secondary problems at the injury site such as cellular debris, scar tissue formation, and there's a need to have the axon reconnect with its distal target, which is often a muscle. So one cue that we're interested in working with is electrical stimulation. The reason we've chosen this is because we did some preliminary studies with a range of electrical stimuli and found that when you apply a field of 50 millivolts per millimeter to sensory neurons, they actually grow bigger. When you apply that same field to co-cultures of Schwann cells and neurons, or the peripheral support cells for the neurons, you actually have an even further increase, which is indicating there's some sort of electrically induced change in these support cells that is then helping to guide this neurite outgrowth. So the study that we wanted to look at next, once we saw that result, was how are these non-neural support cells actually responding to the field in 2D and in 3D, and also are there phenotypic changes in other non-neural support cells, not just Schwann cells, but in the injury site you're going to be having fibroblasts for the connective tissue, endothelial cells for revascularization, and your Schwann cells. So we wanted to look at how those cells also respond to that same field strength. So in order to do that, the first thing that we did was actually make a three-dimensional scaffold. So we seeded a dot of cells, and then we encased that in a matrix, so collagen matrigel scaffold. And then we applied an electrical stimulation using a regime that I mentioned earlier in a custom designed stimulation chamber. So we were able to monitor real time using a LabVIEW program exactly what we're applying so that we know that it's reproducible and all of that. And then after the electrical stimulation with 50 millivolts per millimeter, 8 hours DC, which is what we saw when we did those preliminary studies where the range was the highest response, we then fix it and characterize how far into the scaffold, parallel and perpendicular to the field, that these cells are migrating. The second part of our study was in 2D, and we seeded our cells, so fibroblasts, endothelial cells, and Schwann cells, onto glass cover slips. Then we electrically stimulated them with the same regime, so 50 millivolts per millimeter for 8 hours, and then we let them sit overnight. So instead of putting the neurons on right away, we wanted to see if there's a phenotypic change that takes a little time between the cell types. So we let them sit overnight in new media, and then we added our sensory neurons. So for the results, what we see is if you actually look at these pictures here, this is an inverted image of our 3D scaffolds just from the top. And if you look at the Schwann cell control, which is on the top here, versus the electrically stimulated cells, they actually migrate further into the scaffold. The same trend is seen with endothelial cells migrating further into the scaffold for the electrically stimulated cells. When we quantify this by using measurements in ImageJ software, we get a significant increase in the penetration into the scaffold for the Schwann cells, and we see the same trend with the endothelial cells. However, they're only significantly increased in the direction perpendicular to the applied electric field. When we do confocal microscopy to further look at the actual morphology of these Schwann cells that are migrating further, because they're going to be in direct contact with the neurons, so we want to see what they actually, do they look right, do they look normal that we'd expect. And if you look at the morphology of the cells, in the electrically stimulated samples, the Schwann cells are actually chain migrating and penetrating further into the scaffold compared to the unstimulated controls. So this is telling us that these cells are actually responding to this electrical stimuli to migrate further into the scaffold in a faster manner. However, it doesn't appear to be directional. So it's not just going towards the positive or negative electrical stimulus. It's actually going sort of equally everywhere. So the next part of our project was to look at this in 2D. So how are these support cells that would also be present in the wound, how are they changing, and are they able to further support neurite outgrowth? And what we see is if you look at these pictures here first on the right, endothelial cells that weren't stimulated, fibroblasts that weren't stimulated, and then the electrically stimulated cells on the bottom, you don't really see a change in the green, which is the neurons. They look very similar. But when we do this with the Schwann cells, which are in direct contact in the body with these neurons, if you look at these scale bars, this neuron that's growing on these electrically stimulated Schwann cells is actually much bigger. So these cells are responding and changing. When we characterize this using NeuralLucidus software to actually measure the outgrowth and how many primary neurites there are, branch points and things of that nature, we see a significant increase for the Schwann cells of about 3-fold in total neurite outgrowth and also longest neurite. So they're growing longer and bigger. They're not just getting more bushy and short, which is really important. The other support cells that would be present don't seem to respond to this field strength. That doesn't mean that they don't respond to electrical stimuli, but just that maybe this field strength that's optimized for the neurons in the Schwann cells isn't optimized for the endothelial cells of the fibroblasts. However, if you're going to be applying this in the body to someone with like a peripheral nerve injury, you'd only be applying one field. So seeing this increase in neurite outgrowth is really important. So what we're currently doing is actually going through and we're trying to figure out what are the mechanisms that these Schwann cells are actually responding in. So is it soluble factor release, changes in surface ligand, so what are they actually, how are they expressing this change due to the electrical stimulus? And how can we take that information to further basically either make a new biomaterial or figure out a stimulation regime that would be clinically translatable. So that's what we're working on.
Well, thank you very much.